The alkylamine stimulant 1,3-dimethylamylamine exhibits substrate-like regulation of dopamine transporter function and localization. Abstract. The alkylamine stimulant 1,3-dimethylamylamine (DMAA) is used non-medically as an appetite suppressant and exercise performance enhancer despite adverse cardiovascular effects that have limited its legal status. There is scant research describing the mechanism of action of DMAA, making it difficult to gauge risks or therapeutic potential. An important molecular target of structurally related phenethylamines, such as amphetamine, for regulating mood, cognition, movement, and the development of substance use disorder is the dopamine transporter, which limits the range and magnitude of dopamine signaling via reuptake from the extracellular space. The present studies were therefore initiated to characterize the effects of DMAA on dopamine transporter function. Specifically, we tested the hypothesis that DMAA exhibits substrate-like effects on dopamine transporter function and trafficking. In transport assays in human embryonic kidney cells, DMAA inhibited dopamine uptake by the human dopamine transporter in a competitive manner. Docking analysis and molecular dynamics simulations supported these findings, revealing that DMAA binds to the S1 substrate binding site and induces a conformational change from outward-facing open to outward-facing closed states, similar to the known substrates, further supporting substrate-like effects of DMAA. The drug stimulated dopamine transporter endocytosis in a heterologous expression system via cocaine and protein kinase, A-sensitive mechanisms, mirroring findings with amphetamine. Together, these data indicate that DMAA elicits neurologic effects by binding to and regulating function of the dopamine transporter. Furthermore, pharmacologic distinctions from amphetamine reveal structural determinants for regulating transporter conformation and add mechanistic insight for the regulation of dopamine transporter endocytosis. Significant statement. The alkylamine stimulant 1,3-dimethylamylamine (DMAA) is used as an appetite suppressant and athletic performance enhancer, and is structurally similar to amphetamine, but there is scant research describing its mechanism of action. Characterizing the effects of DMAA on dopamine transporter function supports evaluation of potential risks and therapeutic potential while also revealing mechanistic details of dynamic transporter-substrate interactions. Introduction. Derivatives of phenethylamine include stimulants, e.g., amphetamine, methylphenidate, neurotoxins, e.g., MPP, sympathomimetics, e.g., phenylephrine, antidepressants, e.g., bupropion, and psychedelics, e.g., mescaline. From this broad class of compounds, pharmacodynamic evidence of interactions with monoamine neurotransmitter systems has been used to predict or understand physiologic and behavioral effects and to gauge potential therapeutic benefits or risks. An understanding of the regulation of dopamine, DA, signaling is of particular interest due to the role of DA in controlling aspects of mood, cognition, and movement, and its involvement in a variety of neuropsychiatric disorders, such as substance use disorder, attention deficit hyperactivity disorder, schizophrenia, and Parkinson's disease. Although the mechanisms differ, most addictive drugs increase DA concentrations in their nucleus accumbens, a projection area of DA neurons in the ventral tegmental area. A primary regulator of dopaminergic neurotransmission is the DA transporter, DAT. which is responsible for reuptake of DA back into the presynaptic neuron, since DAT functions at the surface of neurons. Its expression levels in the plasma membrane are critical for DA-regulated behaviors. Consequently, drugs and endogenous molecules can modulate DAT function through direct interactions and by altering DAT trafficking. For example, the DAT ligand cocaine inhibits DA transport but has also been observed to increase the plasma membrane expression of the transporter. Additionally, the DAT substrate amphetamine, AMPH, induces DAT reverse transport but also stimulates endocytic pathways to decrease transporter expression at the cell surface. The molecular events underlying amphetamine-induced DAT endocytosis have been the subject of numerous studies, and there is ongoing debate regarding the trafficking itineraries and molecular mechanisms involved, especially with regard to the role of signaling. In addition to the phenethylamines, there is strong evidence that structurally similar alkylamines influence catecholamine physiology. 1,3-dimethylamylamine. DMAA, methylhexanamine, which resembles amphetamine structure closely, was first introduced as a decongestant. It has since been marketed as an exercise stimulant and appetite suppressant, as have its structural analogues 1,4-DMAA and 1,3-dimethylbutylamine, being included in over 200 products. A number of adverse cardiovascular events led to safety evaluation and, eventually, banning by the U.S. Food and Drug Administration. Although it is difficult to ascertain the current popularity, the substance remains available for purchase and has also been detected in commercial products as an unlisted ingredient. For example, analysis of doping violations from 2003 to 2020 in Norway revealed 9% from DMAA, compared with 26% from cannabinoids, suggesting that DMAA has been and remains a drug of concern for populations seeking positive affective, cognitive, and energy effects of stimulants, especially athletes. Of notable concern, DMAA has been used recreationally in powder form and mixed with other ingredients in party pills. Furthermore, DMAA was found to induce cocaine and methamphetamine-like discriminative effects and condition place preference in rats. Recently, DMAA was found to inhibit the function of the DAT and, to a greater extent, the norepinephrine transporter in a heterologous expression system. Given the sociocultural usage patterns, structural similarity to amphetamine, and emerging pharmacodynamic evidence, we sought to better characterize the effects of DMAA on DAT function and expression. Using a combination of computer modeling and cell culture-based assays of transporter function and endocytosis, we tested the hypothesis that the DAT regulation by 1,3-DMAA occurs in an amphetamine-like manner. Methods: DA uptake assays. HEC-293A cells with stable expression of human DAT, HDAT. pcDNA 3.1-HDAT, our gene plasmid number 32810, were grown to confluence in poly-D-lysine-coated 24-well dishes, and treatments and assays were conducted as described previously in room temperature. 21 degrees Celsius. PBS supplemented with 0.1 mm calcium chloride, 1 mm magnesium chloride, and 10 mm glucose. Following 10-minute uptake of 3-HDA, Perkin-Elmer NET 673, cells were washed twice with ice-cold PBS supplemented with 0.1 mm calcium chloride, 1 mm magnesium chloride, and 10 mm glucose prior to lysis in water with 1% SDS and 0.1 NNaOH and scintillation counting. Background uptake was established by inclusion of 0.1 mm cocaine. Each replicate experiment used both DMAA and amphetamine so that the fold difference between the two could be measured directly within the same experiment. For kinetic assays, DMAA was added 10 minutes prior to and during uptake. Structural model of HDAT and ligand binding. Simulations. Homology model for HDAT in the outward-facing open, OFO, state was taken from the previous work. The three-dimensional molecular structure of DMAA, SID_7753, was downloaded from the PubChem database. Molecular structures of DA and AMPH were adopted from our previous studies. The ligand binding sites and binding poses on the OFO DAT were assessed using docking simulation. Software AutoDock Vena, following the previous approach. In brief, AutoDock Vena simulations were carried out using a grid with dimensions set to cover the entire transporter. The exhaustiveness of the simulation was set to 50, and the algorithm returned 20 binding modes of interest for each conformer. Molecular dynamics simulations of DMAA binding to HDAT. All molecular dynamics, MD, simulations were performed using the NAMD2 software, version 2.13, adopting previous simulation protocol. MD simulation systems were set up using visual molecular dynamics. The control system without DMAA binding was taken from the previous study, in which 2 sodium ions and 1 chloride resolved in the crystal structure of Drosophila DAT were included, and the transmembrane domain of HDAT-OFO was inserted into the center of pre-equilibrated and presolvated 1-palmitoyl-2 oleoyl phosphatidylcholine, POPC, lipid bilayer. The DMAA-bound system was constructed from the control, where the DMAA ligand was docked to the substrate binding site S1. Fully equilibrated TIP3P waters were added to form a box of 104.6 times 104.6 times 150A3. Na and Cl- ions corresponding to a 0.15 m solution were added to neutralize the system. CHARMM 36 force field with CMAP corrections was used for HDAT, water, and lipid molecules. DMAA was protonated using open Babel at the amine moiety as assumed for DA and AMPH in the previous studies. Force field parameters for DMAA were obtained from the CHARMM general force field for drug-like molecules using the web server ParamChem. 2 independent MD runs of 100 nanoseconds were performed following the previous protocol. In brief, prior to productive runs, each system was energy minimized for 50,000 steps, followed by 0.5 nanoseconds constant volume and temperature, NVT, T equals 310 K, simulations and a subsequent 4 nanoseconds Nose-Hoover constant pressure and temperature, 1 bar, 310 K, NPT, simulation, during which the protein was fixed, and constraints on the POPC head groups were gradually released. Subsequently, the constraints on the protein backbone were reduced from 10 kilocalories per mole to 0 within 3 nanoseconds. Finally, the unconstrained protein was subjected to NPT simulations for 100 ns. DAT endocytosis, Ha antibody feeding, assay and image analysis. HIC 293A cells with stable Expression of plasmid encoding DAT with an HA epitope inserted in the second extracellular loop. HA-DAT were cultured on glass cover slips. Cells were incubated with 1 mg per milliliter mouse anti-HA antibodies in room temperature growth medium for 1 hour. Antibodies were removed, and cells were washed 3 times with growth medium and then incubated with 37 degrees Celsius growth medium containing the specified concentration of 1,3-DMAA, amphetamine, or equivalent volume of water. Vehicle. 4. Experiments requiring use of signaling inducer or inhibitor. The chemical was added 20 minutes prior to adding DMAA or amphetamine and remained present throughout. After 30 to 60 minutes, cells were fixed by addition of paraformaldehyde to a final concentration of 4%. After washing in PBS, Psi-3 conjugated goat anti-mouse antibodies, 1 microgram per milliliter, were added for 1 hour to label cell surface. HA-DAT. Following 10-minute fixation with 4% paraformaldehyde and 5-minute permeabilization with 0.1% Triton X100, Psi-5 conjugated goat anti-mouse antibodies, 0.5 micrograms per milliliter, were added to label. Endocytosed and remaining unlabeled surface HA-DAT. Cover slips were mounted on slides using Moeal mounting medium. Z-stacks of XY confocal images were acquired using a spinning disk confocal imaging system based on a Zeiss Axio Observer Z1 inverted fluorescence microscope, with 63x plan. APO-PHNA 1.4, equipped with a computer-controlled spherical aberration correction unit, Yokogawa. CSUX1. Photometrics Evolve 16-bit EMCCD camera, environmental chamber, and piezo stage controller. And lasers, 405, 445, 488, 515, 515, 561, and 640 nanometers, all controlled by SLIDEBO OK6 software. Intelligent Imaging Innovations, Inc. Image acquisition settings were identical in all experiments. For quantitation, 3-dimensional images of at least 4 random fields from each condition were acquired through 561 nanometers, Psi-3, and 640 nanometers, Psi-5, channels. Quantitation of the amount of Psi-3, surface HA-DAT, and Psi-5, surface plus internalized HA-DAT, fluorescence was performed using the statistics module of SLIDEBO OK6. The background subtracted 3-dimensional images were segmented using a minimal intensity of Psi-3 or Psi-5 fluorescence as a low threshold to obtain segment masks number 1 and number 2. Mass number 1 was subtracted from mass number 2 to obtain mass number 3 corresponding to voxels containing only intracellular Psi-5 fluorescence, internalized HA11 complexes with HA-DAT. The ratios of integrated intensities of mass number 3 to mass number 1 were calculated to determine the apparent extent of DAT endocytosis. Similar mechanisms regulate DMAA and amphetamine-induced DAT endocytosis. HEC cells expressing HA-DAT were subjected to the HA antibody feeding assay and quantitative image analysis as described in methods. A. Cells were treated for 15 minutes with 1 mm cocaine as indicated prior to and during exposure to 100 micro-MDMAA. B. Image sets from 5 separate HA antibody feeding experiments were quantitated and collected in a single chart. Each experiment consisted of duplicate cover slips exposed to 10 micro-m of indicated inhibitor or vehicle, no inhibitor, water, or a final concentration of 0.1% DMSO, for 15 minutes prior to and during application of 10 micro-m amphetamine or 100 micro-m DMAA. The ratio of internalized surface HA-DAT in the amphetamine or DMAA condition was set to 1.0 within each experiment. Amphetamine and DMAA did not necessarily yield identical levels of endocytosis in all experiments. Values are means of 6 determinations, and error bars or SEM. Statistical analysis was performed within individual experiments, as opposed to the normalized and compiled data, on all treatment conditions, including the untreated cells. Significant difference from the no inhibitor condition, denoted by asterisk, indicates P and LULT 0.05 as determined by one-way ANOVA followed by the Tukey post-test for multiple comparisons. C. Cells were treated for 15 minutes with 10 micro-m forskolin and 50 micro-m 3-isobutyl 1-methylxanthine, IBMX, as indicated prior to and during exposure to 10 micro-m amphetamine or 100 micro-m DMAA. D. Quantitative analysis of images from experiments in C, which also included treatment with 10 micro-m of the pKa activator dibutyryl cyclic AMP. DB Camp. Values are means of 6 determinations, and error bars are SEM. Significant difference from vehicle, denoted by asterisk, indicates P and LULT 0.05 as determined by one-way ANOVA followed by the Tukey post-test for multiple comparisons. Results were similar in at least 3 replicate experiments. BIM, bisindolyl maleimide I. FIRSC, forskolin. Statistical analysis. All statistical analyses were performed using GraphPad Prism software, GraphPad, San Diego, California. For comparisons of 2 groups, unpaired students' t-test was used after verification of equal variance using f-test. Welch's correction was performed when the variance across groups was assumed to be unequal. For multiple comparison analyses, a one-way ANOVA followed by Tukey's multiple comparison test was used. All experiments were performed at least 3 times. Differences were considered significant when the p-value was an ULT 0.05. Results. DMAA is a competitive inhibitor of the DAT. To test the hypothesis that 1,3-DMAA inhibits DAT function, we measured uptake of 3-HDA by Ishek 293A cells that express the human DAT. 1,3-DMAA inhibited uptake of 3-HDA with a mean IC50 of 29.4 micro-m, minus 14.8 SD, range 12.5 to 66.2, whereas D-amphetamine inhibited uptake with an IC50 of 0.66 micro-m, minus 0.21 SD, range 0.4-0.9, similar to reported values. Representative results from one of 4 replicate experiments overall, DMAA exhibited a roughly 60-fold decrease in potency compared with D-amphetamine across the 4 replicate experiments. 2. Explore the mechanism of transport inhibition further. We examined the effect of 1,3-DMAA on DAT substrate affinity and capacity. In the presence of 100 μm1,3-DMAA, there was a nearly 10-fold decreased affinity of DA for the transporter, with little change in transport capacity. These results suggest that DMAA inhibits DA transport by competitive as opposed to allosteric inhibition of DA binding. DMAA inhibits DA uptake in a competitive manner. A. Uptake of 20 nm 3-HDA by HK293A cells expressing human DAT was measured in the presence of the indicated concentration of D-amphetamine, AMPH, or 1,3-DMAA. Means of 3 replicates, minus SEM were plotted. Drug concentrations for 50% inhibition of DA uptake, IC50, were calculated. B. Uptake of the indicated concentrations of 3-HDA by HX cells expressing human DAT was measured in the presence of no drug vehicle, or 100 micro-MDMAA. Mean, minus SEM of 3 replicates were plotted, and Michaelis-Menten nonlinear regression analysis was performed, providing transporter dopamine affinity, and capacity, Vmax, for each condition. Binding of DMAA triggers the reconfiguration of HDAT from the outward-facing open to outward-facing closed. To investigate the mechanism of 1,3-DMAA binding to human DAT, we performed ligand docking simulations using Autodock Vena. Preliminary docking analysis indicated that the WAS 3S enantiomer of DMAA bound to DAT similarly to D-amphetamine, so that was used for subsequent studies. The most favorable binding site of DMAA bound to the HDAT-OFO was predicted to be substrate binding site S1, which is approximately halfway across the membrane within the extracellular. Compares binding poses and binding coordinating residues of DMAA, DA, and D-amphetamine to the HDAT-OFO conformer. The binding poses of those 3 ligands exhibit high similarities. Their amine groups are involved into attractive electrostatic interactions with D79 via hydrogen bonding or salt bridging, and their hydrophobic moieties interact with hydrophobic residues F76, V152, and F326. Differently, the aromatic rings from DA and AMPH were wedged more tightly between the transmembrane, TM-3 and TM10, via aromatic interaction with Y156, whereas DMAA displayed high flexibility for the binding of its aliphatic moiety. The binding affinities of DMAA, D-amphetamine, and DA to the S1 site were calculated to be -4.3 plus or -0.5, -5.5 plus or -0.3, and -5.8 plus or -0.3 kilocalories per mole, respectively, by Autodock Vena. DMAA binding to the substrate binding, S1, site of OFODAT leads to the closure of EC gates. A. Most energetically favorable binding pose of DMAA to the OFO HDAT, top 2 panels, in comparison with the predicted binding poses of D-amphetamine, AMPH, B, and C, DA2. The S1 site, DMAA, AMPH, and DA are shown in stick format with meshes. Cyan, red, blue, and white represent the carbon, oxygen, nitrogen, and hydrogen atoms, respectively. Interacting residues, making atom-atom contacts closer than 4A with those ligands are shown in the right panel. All 3 small molecules form hydrogen bonding with D79. MD simulations revealed a reconfiguration of HDAT from the D-OFO2 E OFC state after DMAA binding. Time evolution of the distances between R85 and D476, F, and between Y156 and F320, G, in 2 independent runs. Distances were calculated based on the center of mass to further explore whether the binding of DMAA triggers the reconfiguration of HDAT, we performed MD simulations of the HDATOFO conformer in the presence of DMAA bound to the S1 site. Interestingly, within 100 nanoseconds of MD simulation courses, we consistently observed that binding of DMAA triggered significant conformational changes in the EC vestibule, which led to the outward-facing closed OFC, the asterisk designating substrate, and ion-bound state. The OFO and OFC states were distinguished by three major criteria: opening, closure of the outer and inner EC gates, interhelix packing, and hydration pattern of both the EC and intracellular vestibules. First, the F320 side chain underwent a rotational isomerization, enabling its association with Y156 to form the inner EC gate within tens of nanoseconds. The salt bridge of R85D476 that serves as the outer EC gate spontaneously formed, which was further stabilized after the formation of the inner EC gate. The closure of these two EC gates, R85D476 and Y156F320, triggered by DMAA binding, exhibited striking similarity to the sequence of events observed in our previous MD simulations with the substrates DA or D-amphetamine binding to HDAT, which is predisposed to substrate translocation and release as confirmed in our earlier work on LU-T. Secondly, the binding of DMAA prompted an inward tilting of TM1B 10° 15 degrees, and TM6A 2° 10 degrees, segments toward the center of the EC vestibule. The interhelical packing geometry of the EC vestibule reached values typical of OFC state as we observed in the DA or amphetamine-bound HDATO. Thirdly, in contrast to the OFO state where there is a continuous water occupancy in the EC vestibule, in the OFC state, the site S1 is minimally hydrated. DMAA stimulates DAT endocytosis, DA uptake, docking analysis, and MD simulations suggested that DMAA inhibits DAT function and induces conformational changes by binding in a manner similar to amphetamine, albeit with lower affinity. Amphetamine, DA, and other DAT substrates stimulate DAT endocytosis. To determine whether DMAA also exhibits this property of DAT substrates, we measured DAT endocytosis using confocal microscopy and a 2-fluorophore antibody feeding protocol. HEC293A cells stably expressing a mutant DAT containing in HER epitope. Inserted in the second extracellular loop, HER-DAT, carry bound HER antibodies from the plasma membrane to endosomes, allowing efficient visualization and quantitation of endocytosis. And methods for details. HA-DAT accumulated in intracellular compartments following 30-minute treatment with 100 micro-M1, 3-DMAA, or D-amphetamine. DMAA stimulated a dose-dependent increase in DAT endocytosis with a slightly decreased potency compared with amphetamine. DMAA stimulates DAT endocytosis. HEK293A cells expressing HER-DAT were treated for 30 minutes with water, vehicle, or the indicated concentrations of amphetamine, AMPH, or DMAA, and were subjected to the HER antibody feeding assay as described in methods prior to imaging by confocal fluorescence microscopy. Surface transporters appear red and green, yellow in merged images, whereas internalized transporters appear in green only. Single-channel and merged image sets are shown in A for 100 micro-M conditions, merged images are shown in B for 1 and 10 micro-M conditions. Representative. Images from 3 replicate cover slips are shown. C. The fold change compared with vehicle, represented by dashed line, in the ratio of internalized surface transporter was determined by quantitative analysis as described in methods. Values are means of 6 fields of view, and error. Bars are SEM significant difference from vehicle, denoted by asterisk. Indicates P and LULT, 0.05 as determined by one-way ANOVA followed by the Tukey post-test for multiple comparisons. Results were similar in 3 replicate experiments. Next, we sought to determine whether DMAA induces DAT endocytosis by employing similar cellular mechanisms as amphetamine. DMAA and amphetamine-induced endocytosis were inhibited by the presence of cocaine, suggesting that direct interaction with DAT or transport to an intracellular target is necessary for endocytosis induced by both drugs. The role of signaling on DAT endocytosis has been the subject of extensive study. There is evidence for regulation by protein kinase C, PKC, CANI, PI3K, MAPK, protein kinase A, PKA, and Rho-A, with CANI, PKA, and Rho-A being implicated directly in amphetamine-induced endocytosis. Initially, we screened the effects of signaling inhibitors on amphetamine and DMAA-induced HADAT endocytosis. Inhibitors of PKC, bisindolyl malamide I, CANI, KN93, the Rho-A effector ROC, Y27632, and PKA, among other kinases, were examined. Generally, chemical inhibitors should be assumed to be non-specific and are best used to rule out the involvement of signaling pathways. None of these treatments significantly inhibited DAT endocytosis by DMAA or amphetamine. To test the hypothesis that PKA negatively regulates DAT endocytosis, which has been observed previously in cultured DA neurons, we examined the effects of enhancing PKA activity on amphetamine and DMAA-induced endocytosis with the adenylyl cyclase activator forskolin and the CAMP phosphodiesterase inhibitor 3-isobutyl-1-methylxanthine. As shown in this method of increasing CAMP significantly inhibited drug-induced endocytosis. Altogether, these results suggest that DMAA induces DAT endocytosis in the same manner as amphetamine, that is, through a mechanism dependent on transporter interaction that is sensitive to PKA. Discussion. Pharmacologic profiles of many natural and synthetic amphetamine-like compounds are relatively uncharacterized despite their recreational use or inclusion in diet and exercise supplements. Improved definitions of interactions with the DAT and effects on DA physiology can aid identification of public health risks, such as abuse potential due to increased DA transmission. On the other hand, some drugs may carry untapped therapeutic potential, as phenethylamines are used to treat attention deficit hyperactivity disorder as well as substance use disorder, e.g., bupropion for cigarette smoking cessation and prevention of cocaine relapse. Here, we took a key step toward understanding potential risks and benefits of 1,3-DMAA by testing the hypothesis that it influences DAT function and endocytosis in a manner similar to amphetamine. Specifically, we have acquired evidence that DMAA is a competitive DAT inhibitor with an affinity roughly 20-fold lower than D-amphetamine. DMAA binds the DAT and induces conformational changes in a manner similar to amphetamine, and DMAA induces DAT endocytosis by a cocaine and CAMP-sensitive mechanism. The mean IC50 value for inhibition of transport by DAT obtained here, 29 μm, was lower than that reported in a recent study characterizing interactions of stimulants with monoamine transporters and receptors, 106 μm, although the relative decreases in affinity compared with amphetamine were similar, 60-fold compared with 80-fold. In rats, 10 mg per kilogram of DMAA produced discriminative effects similar to 1 mg per kilogram methamphetamine. Reported doses of 1,3-DMAA for performance enhancement or recreation in humans are roughly 10 to 20 times higher than doses of amphetamine used for attention deficit hyperactivity disorder. Dosage of DMAA can be difficult to estimate due to unregulated and illicit sources, as well as mixture with other substances, but seems to range from 25 to 300 mg for oral consumption. Pharmacokinetic analysis indicates that doses in this range result in plasma DMAA concentrations of 0.6 to 20 μm. Studies in rats and non-human primates comparing pharmacokinetic distribution of D-amphetamine in plasma and brain extracellular fluid indicated similar concentrations in both compartments. Since DMAA has similar chemical properties as amphetamine for crossing the blood-brain barrier, e.g., log-P, these results suggest that consumption of higher doses of DMAA can result in acute brain exposure to effective concentrations examined in the present studies. If there is chiral bias for DMAA binding to the DAT, as there is for amphetamine, then the 1s/3s enantiomer of DMAA that was used in the present modeling studies may require a lower dose than the racemic mixture used in the functional assays to achieve DAT-related effects. Physiologic and behavioral effects of DMAA from interactions with other targets should be considered. Importantly, DMAA inhibits the norepinephrine transporter NET, another known amphetamine target, with an IC50 of 0.41 μm. Amphetamine can stimulate NET endocytosis, so it is likely that DMAA mediates biologic effects through interactions with the NET. As well, as DMAA has a greater preference for NET, DAT than D-amphetamine, it may carry greater risks from pressor actions but lower risks from positive reinforcement, limiting abuse potential. In addition to expanding knowledge of DMAA pharmacodynamics, the present studies reveal novel drug-dependent structural changes in DAT conformation. Our molecular modeling and simulations suggest that DMAA can bind to the substrate binding site of the HDAT, with less binding affinity compared with DA or amphetamine, in agreement with uptake data. Hydrogen bonding or salt bridging formed between DMAA and D79 facilitates its binding to the S1 site, which is further strengthened via favorable hydrophobic interactions with F76, V152, and F326. Multiple MD simulations consistently showed that binding of DMAA to the S1 site was able to trigger HDAT conformational changes from OFO to OFC, which may be predisposed to substrate translocation and release as observed for DA binding to HDAT. In the OFO state, hydrogen bonding is formed between D79 and Y156. The binding of DMAA inclined to break this hydrogen bonding via forming salt bridge or hydrogen bond with D79, thus releasing Y156 to form aromatic interaction with F320. We suggest that the amine group in DMAA is important to trigger the reconfiguration of HDAT and, furthermore, that A complete phenyl ring is not necessary for substrate translocation. These findings suggest that other aliphatic amine stimulants, such as 1,4-DMAA, 1,3-dimethylbutylamine, and 2-aminoheptane, also exert biologic effects through DAT interactions. Supporting this theory, 2-aminoheptane interacted with the bacterial DAT ortholog LU-T in silico and inhibited the net in vitro. Investigating DMAA-induced DAT endocytosis may reveal mechanistic details of DAT endocytic trafficking and regulation by amphetamine. DMAA-induced endocytosis is inhibited by cocaine, as has been reported previously for amphetamine. This suggests that interaction with the transporter, or access to the cytosol, assuming DMAA is a substrate, is necessary for stimulating endocytosis. Conclusive evidence for DMAA being a DAT substrate would include cocaine-sensitive intracellular accumulation of radiolabeled DMAA. If DMAA were an inhibitor as opposed to a substrate, it would be the first described to induce DAT endocytosis and would suggest that transporter interactions alone are sufficient to stimulate endocytosis. Alternatively, a proposed intracellular target for amphetamine is the trace amine-associated receptor 1, TAAR1, Ag protein-coupled receptor that is expressed on intracellular membranes in DA neurons. Phenethylamine stimulants have been proposed to activate TAAR1, leading to increased CAMP generation and RhoA activation, with subsequent enhancement of DAT reverse transport and endocytosis. Methamphetamine-induced DAT endocytosis was found to be dependent on TAAR1 expression and pKa activity as suggested by use of the kinase inhibitor H89. However, evidence indicating that amphetamine-induced endocytosis is independent of TAAR1 includes 1. HK293 cells do not express TAAR1, but de-exhibit amphetamine-induced DAT endocytosis, present studies in CAMP and pKa activation, which are stimulated by TAAR1. Antagonized amphetamine-induced DAT endocytosis in heterologous cells and DA neurons present. Studies in cell lines and rodent striatal synaptosomes, pKa activation increased DAT Vmax, consistent with increased plasma membrane expression. Additionally, DMAA induced DAT endocytosis, despite exhibiting no activity at human TAAR1 in receptor binding studies. Therefore, although some evidence does support a role of TAAR1 in modulating amphetamine-induced DAT endocytosis, the present studies suggest that DMAA and amphetamine promote DAT endocytosis through a TAAR1 independent mechanism. If DMAA and amphetamine regulate distinct cytoplasmic targets, their ability to induce similar DAT structural transitions supports a role for DAT conformation in regulating trafficking events, which has been suggested previously using conformationally biased DAT mutations and ligands.